What if we could develop powerful autonomous systems that take on some of the tedium of the trial and error of the scientific method and free up researchers to do some of the more creative work of coming up with bold new hypotheses or connecting the dots on existing data to come up with breakthrough new cures? Well, that's long been the promise of AI and automation, and I got a peek into the state of that in 2026 by visiting the Society for Lab Automation and Screenings, or SLAS's conference here in Boston, Massachusetts. This year's event had over 400 exhibitors, and as I walked through the show floor and talked to folks at booths or at the coffee machine, I saw a cornucopia of fun, whirring machines and robots, but also a couple of key themes emerged from me, as well as two important questions. The first big theme I saw was cross-compatibility. Building a lab is one thing. Getting all of these different instruments from different vendors to be able to talk to each other seamlessly and be well integrated, well, that's a whole other challenge. And so two fun organizations that I saw that were focused on that were first SiLA, a nonprofit based out of Europe that's focused on creating an open source standard that's increasingly being adopted by industry to help all of these instruments communicate more effectively.
I am here with Patrick of SiLA. Organization is doing awesome work in democratizing lab automation. Patrick, tell us more.
So SiLA is a non-profit association that grew out of pain point in pharma companies in Switzerland, where lab scientists, biologists and chemists wanted to do an experiment, and they had trouble integrating lovely different devices in the lab. And you can pay an integrator to connect them, but if you change your mind on the experiment you want to do in the afternoon, you then got to bring them in again and change it. So it's really a bit like a USB for the lab. It's not the cable, it's what sits above it, so that you can send the, instructions to your device and get data back and do your experiment as quickly as possible. Analyze the data and then you're away. New drugs, new materials, your paper, your PhD, and not be frustrated by trying to jam things together. It's hard work.
That's great. I've seen a couple of organizations proudly showing that they are SiLA is compliant and so forth. So can you tell us more about what's unfolding here with other vendors?
So the code, SiLA develops the standard, and we have a community of programmers that develop the code. So the code's available open source, different flavors of programming language. We let anybody use it. Sometimes we don't know that they use it, and then they put it on their brochure, which is fine. Other times we have members, so some member companies like TCAN is part of their integration business. So they're very proud, and it makes their life easier, and it builds more confidence with the customers. So it's a little bit of a link to business strategy, how best to use it. But we want to make it so that an individual scientist, an automator, someone with limited programming skills can just get in there and use it. Open source, lowest barrier possible, get in it, but also interact with the community. So if you have a question or a problem or a request for a feature, you come back and say, hey, I'm looking for this. Probably somebody's done it. And then share, community, open source.
That's great. Well, I met a couple of volunteers from SiLA last night. I feel like this is really the beating part of the effort to make this whole world more accessible to folks, easier to use, and less expensive also to get into. For folks who want to support SiLA, what's the best way for them to do that?
So get onto our website, SiLAstandard.org. You'll find an info at, you'll find a Slack channel, you'll find a link to SiLA Awesome, which is your list of clients and servers, and there's a link into the GitHub. So send an email to info at, get yourself on the mailing list. We are originally from Switzerland, but we love being in the US and love to meet people and exchange and learn what people want to do.
Amazing. And if you're lucky and you get to the booth, you get a nice Swiss chocolate, it seems like, to boot.
So you get Swiss chocolate. We've also got a mascot, Godzilla.
Oh, there we go. That's awesome. Thanks so much, Patrick.
Right, thank you.
Cheers.
On the for-profit front, there was UniteLabs. They are a SiLA-compliant startup based out of Germany that is focused on building the operating system for AI-driven biotech research.
All right, I'm here with Jan from UniteLabs. You all are doing really interesting work in what I think is democratizing lab automation. Jan, tell us more about UniteLabs.
Yes, so we're UniteLabs. We make lab automation more scalable and AI ready by replacing vendor software with low-level control in Python code. And by that, we open up these APIs and make the machines controllable via one central language. And by that, you could argue to say make it democratized to everybody in the industry.
Amazing. And what are we looking at here to your left?
Right here, we have a little work cell that's integrated with UniteLabs, where we have a Agilent Bravo, an ABB robot, Bionoid plate reader, a sealer from Agilent, and everything's integrated with UniteLabs, pure Python code controlling these machines and make a workflow work.
Beautiful. If folks want to get in touch with you, what's the best way?
Just reach out via LinkedIn. I hope you can find me there easily. Maybe we can be linked. Reach out via email. We have our website, unitelabs.io. Just reach out. We hope to talk and exchange thoughts.
Awesome. Yeah, thank you so much.
Thank you.
The second theme that emerged is data generation. In order for these AI models to be working well, they need access to data, a lot of data. And not just data of scientific experiments that have gone really well and that might be published in a paper, but also the many, many experiments that didn't go well. Having that complete picture helps these AI models understand how better to interface with reality, with biology. Well, one startup that's focused on generating these kinds of experimental data sets is called Atinary. They recently set up a lab here in Boston, Massachusetts, and I got a quick tour of their lab and their point of view on this space through one of their scientists.
I'm here with Justin from Atinary. Am I pronouncing that right?
Yeah.
Okay, great. And Justin, what are we looking at today?
All right, so this is the grand opening of our self-driving labs here in Boston for Atinary. So I'm the senior principal scientist here for chemistry. And we've just opened our Boston lab with a couple of autonomous chemistry synthesis systems that are really here to show off the power of automation in self-driving labs and how our software and our company can really run full chemistry reactions in automation, really hands-off from the scientists. And so we have two systems in our lab right now. There's a ChemSpeed system back behind me, and then really the showpiece of this lab is this custom-built system. We partnered with multiple partners, ABB Robotics, Mettler Toledo, JChem, Agilent Technologies, and we have a fully autonomous chemistry synthesis cell with two robotic arms from ABB. We have multiple reactors that run in concert. And on the front end, we can do chemistry, and on the back end, we had it paired with an Agilent open bed and multi-well LCMS system for data analysis, workup, method development. And so really, the scientists can design whatever experiment they want to optimize in our labs. We can run this system looping autonomously, set up the reactions, have our software set up the initial conditions, run the reactions, run the analysis, feed that back into our software, do the interpretation, the analysis, suggest the next set of conditions, start it again, and we can really run this thing in a fully autonomous fashion until we reach the results that we'd like. And so we're really excited to show off this system to show that the power of, that the days of fully autonomous chemistry are here. And we really wanted to show that you can build it right now. And that's what we've tried to do here.
Great. So earlier we were talking about the value of generating AI first data sets and the value of negative data. And that's one of the, seems like, value props of your enterprise and of building these systems. Could you say more?
Sure, absolutely. So, many companies and researchers out there, when they're trying to build their machine learning models and AI models, they train on all the public data that's out there in the literature and everything, which is awesome. There's so much information out there, but the problem is the data, as everybody knows, is very positively biased, right? Nobody publishes bad data or negative results. And so when you want to train an AI model or a machine learning model, it's really important to have a significant amount of negative results as well to really get the full picture. So one of the things we want to be able to do with a system like this is to train our own machine learning models by incorporating and capturing the entire scope of a chemical reaction and the results from the negative all the way through to the positive. And so we think that it would be extremely valuable to be able to capture a much larger breadth of the experiment environment, the landscape, if you will, of an experiment class to generate extremely high quality data that is machine learning readable and can really build much more powerful machine learning models that we can use to improve our own systems, and even make it available for companies to license or purchase to train their own models as well.
Okay, beautiful. Well, thank you, Justin. Really appreciate your demo. And here's to getting a more complete picture of the landscape, as you said,
Yes.
through leveraging technologies like the ones you're building here.
Absolutely.
Great.
The third theme I saw was scale and real-world impact. How do you put all of these pieces together of integrations and automations and AI and robotics and actually move the needle on how science is done? Well, one story that brought that to light was a presentation that was done between Ginkgo Bioworks and OpenAI. And there they talked about how they paired the GPT-5 model with Ginkgo Bioworks' lab here in Boston, Massachusetts, and aimed it at reducing the cost of cell-free protein synthesis. If you want more on that, I recently did a walkthrough of the lab and a deep dive with Ginkgo Bioworks' CEO, Jason Kelly, and you can see that full interview and lab tour linked below. It really does feel like we're in this unique moment for lab automation, one where well-funded startups and governments are putting together a bunch of capital and brilliant people and trying to see if we can create a new paradigm in how scientific discovery happens and how we come up with fantastic new materials or new drugs for illnesses. And like any big bet, it comes with a set of risks. And for me, it raised two big questions. First, North Star for many of the people I spoke to at the conference was around creating an AI scientist, a closed-loop system that could come up with its own hypotheses and its own scientific breakthroughs. And as I think about the incredible complexity of the human body, with its 30 trillion cells and the complex sequential pathways through which diseases emerge, I wonder if there's a ceiling to the degree in which these systems, running on classical computer chips, could ever be able to make fundamental progress to the point where it's able to, let's say, cure cancer. If that's the case, I wonder what is the best allocation of capital between some of these initiatives and others that we know right now need a lot of help, such as human clinical trials that are notoriously expensive and time-consuming. The second is if the promise of AI-enabled lab automation doesn't deliver on the timelines that venture capital firms and that government organizations are anticipating, what will be the spillover effect on some of these smaller startups and labs who are using this technology and actually making meaningful improvements in smaller ways? Could there be a chilling effect in working in or investing in this field, much in the way that there was with AI in the late 80s and early 90s, where a series of overinvestments and hype resulted in an AI winter that took the better part of a decade or two to thaw out? Either way, we're in for an interesting ride. To the teams who are going all in, here's to your success, and to humanity's benefit through new tools, new ways of doing discovery, and hopefully new cures for disease. Thanks for joining me on this tour through SLAS Boston. I'll catch you on the next one.